everyone. So I'm Cristina, and it's really my big pleasure to have you all here and to have Chris with us. So we specially invited Chris here. He's coming from Canada, from Montreal. So we invited him here as he was doing a road trip in Europe. Was going to a conference in Vienna. So we invited him to come over to visit the lab and to meet us all. So Chris, he's a professor at the Department of Medicine in the University of Montreal. And then he's also the director of the Hepatoneuro Laboratory, which is at the University Hospital Research Center, also in Montreal. So I know Chris, I think, since 2012. We were discussing this morning. We met for the first time in Aarhus. So it was at the International Society of Hepatic Encephalopathy and Nitrogen Metabolism meeting. So that was my first meeting, I guess. Yeah. Not really the first for Chris. So based on what the information that I had from his assistant, he's working on the hepatic encephalopathy world since more than 25 years now. Yeah. So, but he's really a young researcher. And it's really a big pleasure to work with him every time. So probably you know also we have now some small satellite meetings from Michelle. So it's a translational Michelle. So we organize every 3 months Zoom meetings, and Chris is always there with us. And then Chris will be also the new president of the ISHAN, of the International Society of Hepatic Encephalopathy and Nitrogen Metabolism. So I think that he will take over now in Germany, end of September, during the meeting, and then he will be the president for 1 year. No, 2 years, because the next meeting will be in 2 years in Montreal at his place. So he will welcome us there at his place in Montreal. So looking forward to meet you in Montreal next time, I guess. So yeah, so it's really my pleasure to have Chris here with us and for him to tell us more about the complexities of hepatic encephalopathy, the role of ammonia, as he's really an expert in this field. Chris, the floor and the Zoom is yours.
Thank you very much. Thank you all for being here this morning, and thanks, Cristina, for the introduction. And as she said, I've known Cristina for, I guess, over 10 years now, if you do the quick math. So today I'll definitely going to be talking about hepatic encephalopathy, which is probably a little bit out of your field. But actually, one of the reasons why I am here, which we've talked many times, Cristina and I, is about collaborations and so forth. And so I'm just going to review sort of the things that we're studying in our lab, mostly model-based. And yeah, and then if you have any ideas, of course, at the end of my talk, I'll be here for a while. So if you want to discuss other things, I'd be happy to. So I'm just going to tell you a little briefly. I won't go into all these details today, but my research program really is translational, in which we work very closely to the Department of Hepatology, which is just across the street at the hospital. So we do a lot of different neuropsych tests in patients. We do biomarkers, we do nutritional evaluation, and we have a lot of animal models, which is important for hepatic encephalopathy. So a lot of neural phenotyping. We do cerebral microdialysis, brain edema, and in vitro we can extract different cells and that are interest to us. But today I'll just go to really focus on one particular point to try and not make the story too confusing, and but it's actually an important topic in the domain these days. But let's start with the story. The story starts with a healthy liver becoming a cirrhotic liver, and it's sort of the insults that induce this cirrhosis or fibrosis is alcohol. Obviously, is still quite strong. Obesity is kind of the next wave, fatty liver disease, which eventually gets to cirrhosis. Viruses, hepatitis A, B, and C. There's a treatment for hepatitis C, so we're seeing less patients with hepatitis C-induced cirrhosis, and autoimmune and biliary cirrhosis. So what really happens is you have fibrosis from 1 to 4, and during the early stages of fibrosis, it's reversible. But at this point, the patient or the person itself doesn't come to the hospital because everything is fine. Because, oh, should I go to the hospital? I feel fine. Then when you get to the later stages of fibrosis and you even get cirrhosis, you get this scarring of the liver. So the liver becomes very hard. Then it's irreversible. And still at this point, the patient is doing really well with cirrhosis because they're compensated. But then all of a sudden they're going to decompensate and a lot of complications arise. And it's these complications that bring them to the hospital. So one of the primary reasons, just because the liver is very hard, you have portal hypertension. That's the the vein that goes to the liver. Because the liver is hard, blood can't pass through the liver, so you get portal hypertension. Obviously, the liver is an important organ for metabolism and even coagulation factors and so forth. So if your liver's not working properly, it's going to cause problems. This portal hypertension will cause vascular hemorrhaging, so internal bleeding of the intestine due to this pressure on all the veins within the intestine. You get ascites, you get an abdomen full of water, encephalopathy, and you get jaundice, which is sort of coloring of the skin. So these are really the reasons why the patient's going to be admitted to the hospital. But encephalopathy is one of the primary causes and most common cause of why the patient comes to the hospital. So by definition is when your healthy liver becomes impaired, has a consequence, and the brain becomes a victim. By definition, it's a debilitating complication of liver disease characterized by cognitive, psychiatric, and motor disturbances. Other ways of saying is that due to liver dysfunction, many symptoms manifest as a wide spectrum of neurological abnormalities ranging from subclinical alterations to coma. And it's important, which is not easy, but this has to be exclusion of other known brain diseases. So if someone has Parkinson's or Alzheimer's, that is not hepatic encephalopathy, but it's very difficult to diagnose. So grading of hepatic encephalopathy, it started from 0 to 4. Back in the day, the West Haven criteria was established in the 1970s, which I think probably did more harm than good. Because if you look at all these different stages and all the symptoms that are associated with each stage, they're very subjective, a lot of overlap. And unfortunately for the FDA, any kind of treatments that were to go through for a treatment of HE, they said, no, this is too subjective. There was an attempt made about 10 years ago to divide into covert HE, which is 0 and 1, and overt, which is 2, 3, and 4. So covert are symptoms which are not very easily to diagnose, mostly memory, concentration. So we diagnose it with neuropsych tests. That's one of the ways we diagnose covert HE. Covert HE is something that's very easy to identify for the clinician. So it doesn't matter what it is, but if it's obvious, then we have overt HE. And we know there is a continuum that patients who have covert HE will eventually develop an overt episode in the next little while. And we call that an episode of overt HE, which brings them to the hospital. And patients who have more than 2 episodes within 6 months, we call that recurrent hepatic encephalopathy. And this recurrence happens a lot over time. So I was fortunate enough to be part of a group of experts, which we recently wrote a review in Journal of Hepatology. I will not go into all the details of what we think is going on in the brain, but that we know that ammonia does play an important role. And ammonia is driven, is risen because of different systemic factors that are occurring within the body. I just want to shout out to Kasia and Cristina that we just recently, hasn't been published yet, but hopefully will be accepted soon, review, which really talks about a brain edema and its association with hepatic encephalopathy. So let's talk about ammonia. Ammonia is produced within the gut primarily. Over 50% of it is produced from protein digestion, amino acid deamination, hydrolysis of urea. So every time we have a meal, we produce ammonia. And then it gets into systemic circulation, it gets to the liver, and the urea cycle is a primary family of enzymes that can remove ammonia. This is just the biochemistry analysis of urea cycle. But ammonia comes in and aspartate, and so this circle just keeps turning and it keeps removing ammonia. But there's also a lot of different enzymes that are used within the body that either use ammonia or produce ammonia. And one is glutaminase, which takes glutamine to form glutamate and forms or generates ammonia. This is found in the intestine, kidney, and in the neurons, especially in the brain. On the other side, we have the opposites, or the mirror image, is glutamine synthetase, which is an enzyme that takes glutamate, removes ammonia, and forms glutamine. So it's sort of the opposite of glutaminase. One generates ammonia, one removes ammonia. And this is found also within the liver, in the brain, particularly in the astrocytes, and in the muscle. And the muscle will be important for later on in my talk. But I mentioned the blood-brain barrier here because Mariana and my lab just recently identified the expression of GS in endothelial cells of the blood-brain barrier. So these are immunohistochemistry showing that in the blue is these GS, which express just in endothelial cells of the blood-brain barrier. So this is a new identification. What its role is really, I'm not sure exactly, but it may be having a role in protecting the brain from ammonia spillover from the blood. So once the liver starts to become severely impaired or cirrhotic, obviously elimination goes down. There is maybe suggested that there's an increased production due to the microflora, like a microbiome changes. In any case, ammonia rises in the blood, gets easily into the brain, and of course it's toxic and causes deleterious effects and cognitive impairment. Now, muscle metabolism is important for ammonia also. It's sort of a secondary defense system. So when the liver goes down, muscle contains this enzyme, which I mentioned before, which takes glutamate and forms glutamine synthetase. And so patients who have cirrhosis and have good muscle mass sort of have a secondary defense mechanism. Unfortunately, patients with cirrhosis have a high rate of malnutrition. About 65 to 95% of patients is involved in malnutrition, which could be many causes. They're not eating enough, impaired digestion, absorption, altered metabolism, they're hypermetabolic. And so alternatively or consequently, sarcopenia arises very often in these patients with cirrhosis. So their second line defense is actually even diminishing even more. And so they're at high risk of becoming hypomonemic and hepatic encephalopathy. So sarcopenia is interesting because these patients are scanned for CT for liver transplant. So there's sort of free information that we can get, and you can sort of evaluate the muscle mass around the lumbar L3 of the spinal cord, which have been already validated to show that it reflects sort of overall body mass. And interesting now, moving forward, is that myosteatosis, so you can sort of evaluate the amount of fat within the muscle of these patients also. What myosteatosis actually means is not really sure. I mean, it does sound bad, but I think we have to look into it at what point it is actually inhibiting or impairing metabolism. So patients with sarcopenia that have less muscle mass have a higher risk of hyperammonemia. So again, coming back to my point that there's a higher risk of hepatic encephalopathy, and these are just sort of a review of a lot of studies showing that patients with sarcopenia have a higher risk of hepatic encephalopathy. So what is really ammonia though? It's a weak acid, it's a weak base. 2% of it is as gas and 98% is as an ion, Na4 plus at physiological pH. What makes it quite special or toxic is that ammonia ion itself is very similar to potassium ion. It has a similar ionic radius, similar diffusion coefficient, and therefore it crosses potassium, it follows potassium across channels and transporters. So basically it gets everywhere in the body, not just the brain. These are all just potassium transporters and channels that are available. There's actually even an ammonia transporter that was characterized a little while ago, which hasn't really gained any momentum, but it was interesting when it first came out. So getting back to my point, ammonia is getting everywhere. And I think that's important to know. I mean, the brain may be one of the first victims because it's a sensitive organ, but there's ammonia in all organs and tissues. So during my postdoc in Berlin, I looked at the fluctuations of pH with ammonia. So this is in a cultured astrocyte preparation. If you apply ammonia to the outside of the cells, you get an acidification of an alkalization of ammonia, of a pH with ammonia, which comes down the baseline. And when you remove the ammonia, you get this acidification. And if you do the same thing with a similar weak base, weak acid with similar 2%, 98% ratio, you don't get this alkalization followed by acidification. So this transport of ammonia going in and out of cells really has a big impact on the pH, which we know has detrimental effects on metabolism. So if you look at the direct effects of ammonia, it doesn't bind to a receptor or so forth. Definitely has an effect on the pH of the system. The fact that it's very similar to potassium, if you flood the system with ammonia ion, you could have an impact on membrane potential. And we know that ammonia is involved in many different reactions. I just mentioned 2 before, but about 30 different reactions that use ammonia or generate ammonia. So if you flood your system with ammonia, that's going to cause metabolic issues. So this is actually the focus of this talk is about hepatic encephalopathy. Is it irreversible? And if you go to the textbooks of all the clinical diagnosis and so forth, that by definition, HE is a metabolic disorder. So it's assumed to completely resolve following liver transplantation. If you fix the problem, everything should normalize. So if we fix the liver, replace the cirrhotic one with a new liver, everything should be fine. And back in the day, liver transplant, as you can imagine, is very invasive. Takes up to 2 hours, 8 hours. And so mortality back in the day was a very important endpoint. But I think we're beyond mortality and survival. So now we're actually shifting to what are these patients doing with the new liver after they go home. And we still see about, in some reports, up to 45% of patients that have been transplanted still have these neurological complications after transplant. So the question is, why? And of course, this is not what we're looking for. But if you take all the patients that had this poor neurological outcome after transplant, I just have to highlight Nicolas Weiss and Dominique Tabou, who recently wrote this review about all the different complications that are seen in patients after transplant. So if you see the ones that had poor neurological outcome, you look into their archives or medical files before transplant, they had a lot of these episodes of overt HE. They were hospitalized many times and so forth. And even if you talk to my colleague, Dr. Bajaj, he says that patients who have these repeated episodes of overt HE become very difficult to treat. They're refractory to the treatment. So there's definitely something going on. So as I mentioned before, patients who are diagnosed with covert HE with neuropsych tests, within 1 year, there's a 30% risk of developing their first episode. Once you had their first episode, there's a 46% risk of developing a second one within 1 year. And that decreases to within 6 months, at 55% risk of having their third episode. So there's something happening with the brain over time with these repeated episodes. It's sort of like the analogy I like to use, because I'm a hockey fan, is a concussion. Once you get one concussion, you're susceptible to having a second concussion and a third. I'm sort of seeing a similar trend with these patients with these overt episodes. So we asked the question, so what are these continuous or repeated episodes of overt HE doing to the brain? So we use a model of bile duct ligation, which you guys also, some of you guys use here. And we've characterized this model for covert HE. So nothing's overt. So you put them in all these different neuropsych tests, these behavioral tests for memory or coordination and gait. And there's definitely a lot of impairment. But if you look at the rat himself, the animal itself, he seems to be okay. So Farzaneh is my PhD student and decided to take on this task. So the BDL model, 6-week model, she injected ammonia at a certain time point in the model, and within 20 to 30 minutes, the animal lost its ready reflex. So animals don't like to be put on the side. For this case, they put him on the side. She stayed there for about 20 minutes. And then it was a reversible dose. So they came up after 20 to 30 minutes also and started moving around. And then Farzaneh did the same thing 4 days later. 4 days later to do 4 episodes of overt HE. And then after about a week, we redid all the behavioral assessments, which we had done, which we are aware of. Obviously, we had the different control groups involved. And interesting enough, that the first episode took a certain amount of ammonia to induce this phenotype. The 4th episode took less ammonia to induce the same phenotype. So coming back to my point, whether there's some kind of fragility of the brain or is more susceptible to this lower concentration is something we don't know, but the observation was very interesting for us. And Farzaneh, just to show you within she did a lot of immunochemistry and looked at different groups, different regions of the brain, hippocampus and cerebellum, and quite an exhaustive analysis. And then what she found in the end is that the hippocampus was something that was primarily affected after these 4 episodes of overt HE. And we saw neuronal cell loss in the hippocampus compared to the other regions. So it was the first time actually we documented that there's actually neuronal cell loss. So if there's neuronal cell loss, you're not going to be able to treat for that. So that could be one of the reasons why these complications persist. And then when you put these animals in the machines, all the different machines, especially short-term and long-term memory, these episodes made things worse in short-term memory and long-term memory. Again, coming back to the point, these episodes sort of make things worse on a behavioral scale. Then we did the proteomics and for those who have definitely have used this technique before, you get a lot of data. So this advantage analysis, which takes into consideration all the 4 groups, we've identified a few proteins which I'm not 100% sure what they do, but we're trying to see whether we can pursue something a little more specific in the future. So, and then the other point, I wrote a grant some years ago asking the question, what about someone who has hepatic encephalopathy at the time of liver transplantation? So we talked about the effects of all the episodes, but what about someone at the time of transplantation? My hypothesis was based on the fact that transplant, there's a lot of perioperative conditions. I mean, there's a lot of the time of the surgery could be 2 to 8 hours. There's a lot of blood loss, a lot of hypotension. There's anesthesia, and even after transplant, these patients are on continuous immunosuppression, which we know can be toxic. So my whole point is, someone who goes into transplant with a HE brain, I'd like to refer to that as a frail brain. So a compromised frail brain becomes predisposed to what would normally be innocuous perioperative insult, resulting in cell injury and cell death. So what we did, we took these BDL animals again, and we have normal rats there, and then we have our cirrhotic rats with covert HE, and we looked at neuronal cell loss following hypotension in both the sham and the BDL, and we saw a decrease in neuronal counts in the BDLs that underwent the hypotension compared to the shams who went also through the same hypotension. But then when we do these, we weren't sure whether it's the cirrhosis or whether it was the covert HE. So we treated these animals with ornithine phenylacetate. So as you can imagine, one of the targets for treatment of hepatic encephalopathy is lowering ammonia. And OP, I'll just refer to ornithine phenylacetate as OP, just passed its phase 2B study. So it's an interesting molecule which takes ornithine and phenylacetates, and ornithine is used to go inside the muscle, which is actually stimulating the enzyme, which I was talking to you before, glutamine synthetase, removing ammonia and forming glutamine. Now, glutamine also, mentioned before, can find glutaminase, which I mentioned the enzyme before, which can regenerate ammonia and reform glutamate. So all the ammonia removed here can be reformed here. So your balance at the end is not really the best. But phenylacetate is actually a treatment which has been given to children with UCD, urea cycle disorders. Phenylacetate is given, which is going to combine with the glutamine that you form here. In theory, it's going to form phenylacetylglutamine, and that cannot be metabolized by glutaminase. So everything you're removing here is actually formed with phenylacetylglutamine, and it's excreted in urine. That's the whole hypothesis and mechanism of action of OP. So when we gave OP to our animals, we saw a positive effect on blood ammonia levels, and then we looked at different neuropsych tests, and we saw that OP did protect the brain from any kind of neurophenotype impairment. And then we redid the studies again with hypotension. Again, the patients and the animals that were treated with OP, they were protected against this neuronal cell loss with both the 2 different markers that we used. So my whole point is that an HE brain, of course, is frail. And if you look at even hypotension, we just recently wrote this review describing the impact of hypotensive insults and chronic liver disease. It may not just be ammonia, but it also could be the hypotension. And so if you look at patients with cirrhosis have very circulatory abnormalities, and even the GI bleeding and precipitating events of HE involved hypotension. And so we're wondering whether hypotensive should be actually a little more closely monitored and maybe be identified as a precipitating factor of HE. So ammonia toxicity, as I mentioned before, involved a few editorials about ammonia's getting everywhere, maybe also in causing problems for other organs. So you dive a little deeper into the research or the literature about how ammonia can impact the kidneys, the lungs, and the heart. There's not really great studies, but a lot of anecdotal evidence demonstrated that ammonia can play a role. David Shawcross from the UK showed that neutrophil bursts are impaired, affected with ammonia. And even Rajiv Jalan from the UK is trying to describe that ammonia can accelerate fibrosis, especially in the model of NAFLD and fatty liver disease. But there's been a lot of work, and I have to tip my hat to Dazaratsi from the US, about the impact of ammonia on muscle. So we looked at sort of the metabolic reprogramming of hyperammonemia and its impact on muscle. So the sarcopenia that we see can be could also be induced by hyperammonemia. So we decided to take on this this challenge, and we took the BDL models, and we chronically this BDL model also has been characterized recently in, well, for us, 2017, that there's loss of muscle mass in this animal and there's a decrease in protein synthesis at 6 weeks. So we decided to give OP from day one, or week one, for 5 weeks. And we just recently submitted this article demonstrating that if you lower ammonia with OP chronically for 5 weeks, you prevent this muscle mass catabolic state, and there's preserves muscle mass and muscle function. So come back to the point that maybe if we treat hyperammonemia from the get-go, may not only be protecting the brain, but we could also be protecting the muscle. And so muscle, as I mentioned before, is the second line of defense. And so a lot of people are targeting the muscle. How do we maintain muscle mass in these patients? And exercise is the most obvious treatment to say. So there's a lot of work done with aerobic exercise and resistance exercise. And actually we did a study in our animals looking at progressive resistant training in our animals. You may ask, how was that done? But so we have this sort of fence. I had a student from Denmark study. She devised this fence where the animals can crawl up, and there's a weight attached to their tail. She'd do every day different training, and she'd increase the weight in the animals. And she did it for the whole 5 weeks. It was a really great study. And she showed that if you did enough exercise, especially resistant training, that you could maintain and prevent this muscle mass loss. So conclusions: covert to overt, recurrent to irreversible, I think is something that has to be well respected. I think in clinical practice, the whole issue, but don't worry, given the liver, everything will be fine. I think that has to change. I think episodes of overt HE can render the brain fragile, therefore vulnerable to any innocuous insults that may happen along the way, leading up to transplant and even past transplant. I think episodes of overt HE can lead to irreversible brain injury, but I think there's a little more, we have to get a little more granular with details. As we look at the episodes they had before transplant, it's important to know how many episodes over time, how long was the overt HE episode, not just as an episode. I think these are important questions that we have to ask. And so we've sort of declared that from a metabolic disorder to a neurodegenerative condition, you go from minimal covert HE to overt HE, different episodes. Then they go through transplant. They face all these sort of perioperative factors. And if they get out of transplant, there's also these post-LT factors. So it's a rocky road for the brain from fibrosis all the way to past transplantation. And we know that there's a lot of complications that could arise after transplant. Again, these complications we see after transplant, they could be a synergy of these along the way, or one, or so forth, which we don't know at this point. I think ammonia is definitely a neurotoxin and can impact, obviously, brain function. But I think it's safe to say that it's also a body metabolic stressor. I think overall, ammonia can play a big role. And so the question is, do we treat hyperammonemia independent of whether the patient has HE? But for me, it's very similar to glucose with diabetes, or we treat glucose levels whether the patient has any kind of symptoms. I think that's the way we should move forward with hepatic encephalopathy. We know muscle plays an important role in ammonia metabolism, so it's become a therapeutic target, either to maintain muscle mass or to use treatments that can stimulate ammonia metabolism in muscle. I think in the end, the best thing is to try and prevent these episodes of overt HE in patients. So if ever they are transplanted, they'll have a better chance of a better neurological outcome following transplant. This is my group this past Christmas, which I have to thank them because they make my day more joyful than ever, and a great gang. Really stimulating, and we have a lot of good discussions, and it's good to be with them every day. Thank you very much. Any questions, please?
I don't know if you have questions, but while you're thinking of your questions, I might have some.
Like?
We saw in our BDL rat that cerebellum shows the strongest changes.
Mhm.
And it's quite interesting to see that. So we have the strongest glutamine increase in the cerebellum compared with, then it's hippocampus and striatum, for instance. Then we saw osmotic, the release of the osmolytes due to this glutamine increase, which is quite different in the striatum, which is very strong, knowing that glutamine is not so strongly increased. Then the antioxidants are different in the cerebellum. And I was not curious, I was surprised to see that when you go to with the BDL rat, but when you induce hyperammonemia even stronger, so you create a acute on chronic or
Yeah, it's not really acute on chronic by definition. More for me, it's more of an episodic. It's decompensated. Yeah.
Okay, you see that hippocampus has the stronger neuronal loss.
Yeah.
It's quite interesting to observe this and at the beginning, so nobody knows today what's the brain regional differences in HE, or we suspect some, but we never really looked into them. And we have some data and probably will be publishing them soon, or submitting them soon, probably. And there's a difference between submitting and publishing. It was done now with the power, and it's quite easy to publish them before they're accepted. And it's still an interesting topic. So it would be nice if we could look into it further on, because it really seems that brain reacts very differently. Every brain region reacts very differently. It's just chronic liver disease, it's episodic HE with chronic liver disease. Maybe have patients.
Yeah, I was just referring to mostly neuronal cell loss.
Yeah.
I mean, whether there's alterations in other regions, it's very possible.
Yeah.
But as far as neuronal cell loss, we found only primarily in the hippocampus. I guarantee the other regions are not doing very well also. Yeah. But at this point, at this time, there's no neuronal cell loss. Maybe going later, they would eventually have neuronal cell loss. But so a little bit different in yours. I mean, your glutamine peak is not meaning that there's neuronal cell loss. Of course. It means there's things are not going well, which.
But it's puzzling me, or it's really a nice puzzle to look at, to see what's happening. You just start with the model of chronic liver disease, you just leave it as it is, or you just induce something.
Right.
And it seems that if you look at neuronal cell loss, there's differences between brain regions. If you look at how glutamine and the osmotic stress reacts, it's again another difference, and it's not the same.
No.
It's different ones. So that's quite an interesting aspect to
that's true.
Look at.
Yeah.
Maybe I have a comment and a question in comment together. Exactly concerning this, what you're talking right now, because cerebellum and hippocampus, they're completely two different structure. Cerebellum, you have myelinated axons, whereas in hippocampus, 80% of the axons are unmyelinated, so they are less protected. And the blood circulation is much lower within the hippocampus than in cerebellum. So then maybe also the evacuation of the ammonia, it's lower at the hippocampus. That's why those cells are more exposed in the case, to become, to the necrosis of the activation of the death.
Yeah, it's been found in other neurological diseases that hippocampus is kind of a primary sensitive region. Exact reasons could be, as you mentioned, could be regional blood flow or so forth. As far as whether ammonia gets out slower or quicker, Anna did she did a study looking at the evacuation of different factors, not ammonia, but
I don't remember.
But ammonia will follow its gradient, so it doesn't have to be. That doesn't have to be discharged. It will follow its gradient. So if ammonia goes down, it will come out. If ammonia goes up, it will, and so forth. So I don't think ammonia evacuation is slower. If the blood is the same in all brain regions, then ammonia exchange will be the same, I find.
Now, then follow up on the transplantation. We shouldn't wait for the over then with the patients. We should then think about the liver transplant.
That's a very good question.
Yeah.
I've actually wondered whether overt HE should become a contraindication.
Yeah.
But right now it pushes patients to get transplanted. Mortality is important, so the patient will definitely survive longer, which is also important. But to whether that patient benefits best from that liver. There's a lot more patients waiting for livers than livers that are available.
Yeah.
So the criteria, selection criteria is pretty important. But yeah, I could see. Not sure if number of episodes can be part of this diagnostic or prognostic scores for liver transplant. You know, the MELD is used to see who's going to get a liver and so forth, but maybe we can somehow integrate the HE into the MELD score. Must be any dumb question. I take any. They're not dumb questions. Naive questions. You guys are not in the field, so if you have a question, please, I'd be happy to.
Can you maybe comment on the sex difference? I saw your recent presentation.
Yeah. I didn't mention it today, but we did female BDLs, and which react a lot different than the male BDLs. So we saw that the male BDLs lose muscle mass, the females don't lose muscle mass. And the ammonia levels, I think, a little bit lower. Yeah, we just published that article, and we haven't done it with anything else besides that. It's been talked about whether we should induce these episodes in the female to see whether they'll become also reactive or not. But I think we're going to. I don't know who's going to. Those students come forward saying, "I want to do that project," but it would be interesting because I think they'll react differently, which would be a great observation.
I was also, it's the first time that I really see a nice presentation of the OP treatment and that I really understand what's OP doing. Now the fact that, it's taking ammonia and converts ammonia, and that also takes care of the glutamine.
Yeah.
And we discussed once with Rajiv, because it's Rajiv who was inventor or founder, whatever, And we discussed once, we even had some OP here in the lab. Anna left it when she went back to UK. And then we had to ship it back to them because they had a problem. So that's a nice story, by the way. But that's quite interesting, because if it's lowering ammonium, of course will lower also brain glutamine, but also might also help the brain glutamine to decrease even more, not only by ammonium. But the question is, what do you do with, you have the glutamine, so you have the ammonium which forms glutamine. So with the treatment, you will be taking care of the newly ammonium created, but what do you do with the old glutamine which is there? So maybe OP will also help on, I don't know how we can break the two things apart, if we should break them or not, but it would be nice to look at
You're aware of it. The children with urea cycle disorders, glutamine is a big problem with those patients. Not so much in the brain, but there's some evidence, but so they're just given phenylacetate because GS, glutamine synthase, they don't have liver failure. Urea cycle problems. They have a lot of glutamine, so they're just given the phenylacetate to remove the glutamine. Whether phenylacetate gets into the brain, that'd be a good question. And if it's removing the glutamine in the brain by phenylacetate, that'd be good. So you just have to give phenylacetate.
to see what's happening.
You can separate orange.
To separate the two of them, yes.
Yeah.
We do it together. Yes. Any other questions?
Just a curiosity, does the primary cause of the hepatic encephalopathy change the outcome of the patient? Because I think that if the hepatic disorder is caused by alcohol, for instance, alcohol also already has an impact on the brain, while a hepatic virus does not impact the brain. So,
yeah, no, it's a very good question. And it is difficult with the alcohol because we know alcohol has definitely an effect. And I guess it comes back when you do these neuropsych tests. The problem with neuropsych testing is that when you do one patient, it's a snapshot. You're not sure if that patient was like that before they had cirrhosis or not. So alcohol, there's no doubt alcohol plays a role, whether it becomes, and then it's again a second degree of susceptibility. Alcohol brain may lead to higher risk of HE brain. So sort of another fragility, but it's difficult to study and it hasn't been studied very much. I mean, alcoholism and so forth is a whole different domain, but as far as alcohol relating to HE and in cirrhosis, that's not very well studied. But we just did a model recently where we binge drinked. We gave alcohol like a batch of PDLs for the whole 5 weeks. And then we're trying to see, we haven't finished the analysis yet to see whether that impacts something at the end. So BDL with alcohol versus BDL without alcohol, so let's see what it affects. But that's a really good question.
Any other more questions? Yeah. Thank you. Thank you, Chris.
Thank you.
What was that?